So, the phospholipids are amphiphilic molecules. They are composed of phosphate moiety and the fatty acid moieties. They are synthesized in the smooth endoplasmic reticulum, and then they move from SCR to the Golgi complexes, and then they take their final route from there either to the plasma membrane, where they become a part of the plasma membrane, or they move outside through the process of exocytosis. Then there are also phospholipids, which are ether containing phospholipids. We have already told you some very important phospholipids like plasminogens are very important ether containing phospholipids, and the phospholipids involved in the inflammatory or inflammation based responses. They are also ether containing phospholipids. They they begins their route towards the cell membrane or exocytosis in the peroxisomes. Phospholipid synthesis, specifically the glycerophospholipid synthesis, is a very interesting topic. First of all, there are two ways through which the glycerophospholipids can be formed. For instance, one is the donation of a phosphatidic acid from CDP based or CDB containing diacylglycerol to an alcohol, which give rise to the formation of glycerophospholipid. You know that CDPs are involved in the activation of intermediate compounds, which then leads to the accomplishment of the making of the final product. So that activation through CDPs, or basically from CTPs, which give rise to CDP based conjugated molecules, is very important. So one way is that you get a phosphatidic acid from CDP diacylglycerol, which is then donated, which is donated to the alcohol, giving rise to glycerophospholipid. Then there is a second route also, second or alternative way of making glycerophospholipids, the donation of a phosphomonoester of alcohol from CDP alcohol, from CDP alcohol to diacylglycerol. So this donation of phosphomonoester of the alcohol from the CDP alcohol to diacylglycerol also give rise to the formation of glycerophospholipid. So these are the two ways through which you can get the glycerophospholipid. So the major thing here you have to keep in mind is the CDP based activation of the intermediate compounds, like of the diacylglycerol, for instance, or of the alcohols. And this activation, why this activation is required, we have going through this again and again, that this activation is important because it provides the energy for the reaction to get complete. Then there are different examples of the glycerophospholipids which are synthesized and then utilized. Two major examples are phosphatidylcholine and phosphatidylethanolamine. The phosphatidylcholine and phosphatidylethanolamines are synthesized in a very simple transfer based pathway, in which, for instance, the phosphatidylserine, again a sort of a glycerophospholipid, it is decarboxylated, and through the action of the transferase enzyme, it gives rise to the formation of a phosphatidyl ethanoyl amine. So first is decarboxylation of phosphatidylserine occurs, and then ethanolamine moiety, which you get from, let's say, from the diet, and or from the degradation of already present what we say the phosphoglycerolipids, and this gives rise to phosphatidyl ethanolamine. So two enzymes are involved, again decarboxylase and then phosphatidyl ethanolamine transferase. Once we get the phosphatidyl ethanolamine, it gives rise to the formation of phosphatidylcholine through the activity of N-methyltransferase enzyme. This N-methyltransferase enzyme, actually, what does it do? It helps in the transfer of the methyl moiety from S-adenosyl methionine. So in this way, because of this transfer of the methyl moiety, you get, and of course, there is a combination of different reactions, but for the sake of simplicity, we are showing you the major reaction which leads to the formation of phosphatidyl choline from phosphatidyl ethanolamine through the action of N-methyl transferase. Also, you should keep in mind that a phosphatidylcholine uh, is produced, like many other phospholipids, is produced in the liver, and it's very important part of phospholipids. And its importance can be seen from the fact that even if the free choline levels are low, still it is required, and its formation goes on. I mean, getting it goes on being formed because of the requirement of the body of this important compound. Then finally, I have told you that phosphatidylcholine and some other phospholipids they also act as the surfactants, specifically in the lungs. So what surfactant do? That they make the lung surface a little bit more, I mean, they decrease the surface tension when the lung is undergoing different cycles of expansion and relaxation, expansion and contraction. So as a result of this process, there is always tendency of getting more and more rigidity, and that rigidity, that surface tension is decreased because of these phospholipids. One of these phospholipids is DPPC, and which is a dipalmitoyl phosphatidylcholine. So in this way, the very important organ, one of the most important, I would say, depends for its function on this relatively small molecule but very important molecule. And also the composition of the surfactant, that how much lipids it contain and how much your lungs have it, it also acts as an indicator of the maturity of the lung of the fetus. Of the That's why it's act as an indicator of the fetal lung maturity.